guys. We are going to finish skin and start on nervous system today. It's not the hard part of nervous system. That will happen right after your lab practical. Actually, we'll probably get started before then, but you won't be tested on it until after your lab practical. So you have a quiz due tomorrow. Yes? Make sure you get it done. Right? Eventually this semester I'm going to stop reminding you of the quizzes because you're going to be a grown-ass adult.
No.
Yeah, it's totally overrated, but I don't want to hear it either, so let's do it together. Okay, and then how are you guys doing in lab? Is it starting to click at all? What? It's getting wild. It gets a little chaotic in there. Yeah. Is it starting to sink in?
Yes.
Yes. Good. It's getting better, right? You're not. Huh? I mean, except for you, right? It's not. Is it starting to sink in for most of you? Where's Mazzy? You are her keeper.
Yeah. It's exhausting.
You're like, I have a husband and a cat. I don't need you. Any issues, questions? Everybody okay? I think that a couple of the lab instructors are going to set up mock practicals during. Go ahead.
This is a question more for our lab instructors, but when they talk about insertions and all that, are they like super keen on the exact insertion of the heel, just the bone? Okay.
Yeah, you will not get credit if you just say humerus when it's the deltoid tuberosity of the humerus. Gotcha. You won't get credit. Okay, go ahead.
What about like weekly doing something like for actions? There's ones that are like weekly flexes versus.
I don't usually make people say weekly, and they don't usually either. It usually just means that that's not the primary mover. It's not the agonist, right? It's a synergist, right? So they don't usually hold you responsible for weekly, right? It's just me trying to tell you that, for instance, the temporalis isn't a major elevator of the mandible, it's the masseter. Does that make sense? Okay. So, but one last little thing and then we'll move on, and that is that you are all going to be tested relatively similarly. Where is it? And that is, did you see the breakdown that's at the top of SLCC anatomy? Did you see it that shows you how many questions over each thing? Okay, if you didn't, you should, because of course eventually you're going to want to plan your time. And it is right here. Okay, approximately 8 questions on anatomical terminology, 12 questions, ID, functions, locations, features, tissues, 26 questions on bones, features, size, significance, and muscles, 24 questions, muscles, attachments, actions. And then here's how the spelling process works, here's how the attachments work, the parenting, the actions, one action, we all agreed. Everybody's asking one action. You have no idea how much freaking blood was spilled in the process of trying to get them all to do it the same damn way. So they are going to do it the same way. What?
Just one action, not all of them?
Nope, one action.
And
Yeah.
one insertion?
Yes.
Oh, good. Okay.
Yep. That's not for me. I will ask you anything I damn well please.
Right.
Okay? But that test is not the same week, it's the week after. Yeah? It's over mostly the same stuff, Bridget. It's going to be, like, I did it on purpose. It's over muscles, right? And it's over skin. And you haven't done nervous system 1 yet, but you actually don't do that in lab anyway. So that's basically just sort of the nervous organization, okay? So there's a lot of the same stuff that's on that test. Everybody okay? Any questions? Any issues? Yes? No? Okay. So one last little thing, and that is that as you start to get really stressed about your lab practical coming up and you think you might want to skip lecture, I want to remind you that lecture is 75% of your grade and lab is 25%. So don't be bad at math and skip lecture. And if no one comes to lecture, I will not post those videos. Do not think I will. I will not. I'll just keep them on my computer until the day that I think I have an extra few minutes to post them, okay? So, because that's extra anyway. That's gravy, isn't it? So, and those of you who are here will be like, I don't care, I was there. Okay, so let's finish up the epidermis, do the dermis. We'll finish up the skin. I've got some weird, cool crap to tell you today, just because I got bored and I did a whole bunch of reading on skin stuff, and I like weird stuff. So let us. I think we did stratum granulosum last time, right? And lucidum, didn't we? Did we do granulosum and lucidum, just those two layers? There's the granulosum shown really well. And then the lucidum is right there. The lucidum, you don't have to ever label it in thin skin, right? We said that. You don't have to label the lucidum. And then the last layer of this is just the stratum corneum. And the stratum corneum, as in corn or horn or unicorn, is really just a bunch of cell membrane bags filled with keratin. And it's all dead by the time you get up there, right? And so this is thick skin and this is thin skin. So this might be like the palms of the hands, and this might be the skin around your eyes, right? Okay. But this primarily gives you both waterproofing and protection from mechanical abrasion, right? That's what that layer does on the top. Now, this isn't more cells to, for instance, give nutrients to, is it? No, because they don't need anything anymore, right? So this is the living portion of the tissue, and this is the living portion of that tissue. Does that make sense? So keep that in mind for when we talk about blood flow just a little bit. So now, one last reminder. The reason that when your skin is dry, for instance, and you scratch it, you can see some skin flake off sometimes, is because when all of those keratinocytes or basal cells were made originally, they were all stuck together with desmosomes, and as they move up, 30 days later, not all the desmosomes have broken. So whereas you can't see a single cell with the naked eye, you might be able to see a cluster of 300 with the naked eye. So that's why you can see skin, sort of flaking off when it gets really dry.
What happens with psoriasis?
Psoriasis is immune system response on your skin that actually causes irritation, and therefore your rate of skin cell division increases because of the irritation, right? That's all it is, really. And there's genetic predisposition to psoriasis, and there's environmental irritants that can cause it as well. Okay, so that's the stratum corneum. And now let's just do the dermis. Okay, now we are moving down, and here's your dermis. And it goes from here until you start seeing a decent amount of adipose. That's all the dermis. It's got two layers. The papillary layer, which is made of what kind of tissue? Yes. Areola connective tissue. And unless you have a really good histology slide, it doesn't really look different. But it is. Primarily, it's just got fewer bundled collagen fibers. It's got a few more elastic fibers. It's a little more stretchy than some reticular fibers. But again, you can't see the difference here. It's the thinnest portion. It's right next to here, right next to the epidermis. And then the reticular layer is not called reticular because it has reticular connective tissue. It is actually what kind of tissue in the reticular layer? Dense irregular. So why is it called reticular? Because reticular doesn't just mean reticular fibers. Reticular, like the endoplasmic reticulum or reticulated python, means curvy. Yeah? So those really curved, dense irregular connective tissue fibers down there is the reason it's named that. Not because it has reticular fibers, right? And reticular fibers are called reticular fibers because they're really curvy. So don't let that kind of confuse you and make you think that there's reticular connective tissue. So this is, of course, the thickest layer of the whole skin. And it has a whole bunch of other organs in it. It's got sensory receptors and erector pili muscles and sebaceous and sudoriferous glands. Whole bunch of blood flow in here. The dermis, of course, is responsible for nourishing the epidermis, which is avascular, yeah, from the bottom to the top. But again, it's possible to do this because the epidermis is microscopically thin, even in thick skin, because the thick is not because the epidermal living portion is thicker, it's because the dead portion is thicker, right? So you can still get oxygen and nutrients all the way to the top via diffusion, and waste products and carbon dioxide all the way to the bottom via diffusion. That makes sense? So it's still good at doing that. It's really, really vascular tissue. So highly vascular, lots of nerve fibers, touch temperature, pressure, pain receptors in it in the dermis. There are lymphatic vessels. They're not labeled here. Lymphatic vessels are primarily involved in both draining excess tissue fluid and the immune system. They don't really show any of them in here though. And then erector pili muscles, and then the contours of the skin where the epidermis meets the dermis is not smooth, right? So these are called dermal papillae. And if you pull them apart, it kind of looks like this. Dermal papillae, do you see those? Papilla means nipple. Every single time they found anything that was vaguely nipple shaped, they called it a nipple, right? Okay, because it was a bunch of Catholic monks who were naming all this stuff. So, and they were like, I'm pretty sure that looks like a nipple. So you got a nipple in damn near every body system. So it makes a stronger association between the epidermis and the dermis. That should make sense to you instead of a nice smooth association between them. So those are called the dermal papillae. In some regions, hands and feet, we have evolved really dramatic ridges between the dermis and the epidermis so that the epidermis follows the dermis. Yes? And those are called either epidermal ridges or dermal ridges. And they manifest as fingerprints and footprints. Evolutionarily, what's the advantage to fingerprints and footprints? Yeah, these are also called friction ridges because holding on to things with them or stepping on things with them is kind of like the difference between wearing your church shoes and wearing your hiking shoes. Which one are you going to land on your butt in? Definitely your church shoes, right? Okay, so these are genetically inherited. They're vary from person to person. They're really, really useful as identifiers of people. You guys know this, right? They've been doing it since the 1800s. If you have siblings, your friction ridges, dermal ridges are going to be more similar to them than they would be to me, right? Anybody in here got an identical twin? No? Huh? Fraternal. She's just like a sibling. She was just like roommate from the very beginning is all, right? But identical twins have, of course, identical genetics, right? It would make you think that they would have identical fingerprints, but they don't. They actually have really similar fingerprints. Their genetic predisposition to fingerprints is identical, but they are impacted by the friction that's occurring when they're in the amniotic sac and during development. So, for instance, if one of the twins developed with their fingerprints kind of pressed up against their body or pressed up against the wall of the amniotic sac, then it changes the development of their fingerprints. So you can actually tell them one from the other. They're super duper similar to one another. So every single time I have an identical twin, I'm like, please bring me your fingerprints. Because they're so similar to one another, but they're actually distinguishable. Which is kind of interesting because, of course, with DNA fingerprinting, which isn't fingerprinting, it's just looking at your DNA code, I can't tell the difference between identical twins. So if you got an identical twin, you literally always have an alibi, right? Or at least somebody else to blame. You're like, I'm pretty sure it wasn't me. Prove it. So this is the nature of fingerprints and footprints. Okay, so let's talk about tattoos. So if, of course, you did a tattoo and you only put it into the epidermis, how long would it last maximally? A month, right? It lasts a month, okay? Because you get a complete turnover. So that would be like a henna tattoo, right? That you get when you go to Mexico, or I got one in Thailand. If you get a really cheap one, it'll probably be gone in 3 days, right? The one that I got in Thailand lasted 2 weeks, but still, you know, exfoliation is going to take off. So what's the difference between that and a real tattoo? And real tattoos have been around for hundreds, if not thousands of years. And what happens with a real tattoo is that you inject the dye all the way into the dermis. Yes? That's a red tattoo. And what happens is your immune system walls off that little ball of dye, saying, you're not poison, and they figured out the tattoo dyes so that, generally speaking, they don't irritate a whole hell of a lot of people's skin now. It walls it off. It says, I don't know what you are, but if you stay right there, I will not whoop up on you. So your immune system sequesters the tattoo dye, which is what makes it stabilize and stay in that position, okay? And generally speaking, it's going to have a decent amount of longevity, especially since we've developed a lot more tattoo dyes than we had historically speaking. You know, historically speaking, they just used like soot, right? And just banged it in there. But now we have tattoo dyes that are relatively stable, less likely to cause a negative immune system response, and UV stable. That's really important, that they don't break down with UV radiation as quickly as other dyes do. So that one goes in there, and then if you keep it stationary, then you don't have a whole lot of bleeding, and you can get a lot of detail on a tattoo. Does that make sense? Okay? Now, I know some of you guys have tattoos because they're really common now, right? Pretty common now. So how do you make sure that your tattoo has some longevity to it?
Sunscreen.
Yeah, you have to take care of it with sunscreen. They break down with UV radiation really dramatically. And then if you don't take care of it initially after the damage, and by the way, you have to damage the skin, right? Like that's literally what you were doing. And so it's not going to be super comfy, right, to get it in there, right? Especially depending on where you put it. So you have to damage the skin. And what some people don't do is they don't take care of it immediately after. And if you let inflammatory situations occur, then you are going to bleed the dyes and you are going to lose those crisp lines. Does that make sense? So you have to take really good care of it until it heals, right? And then if you want for the dyes to last a long time, you have to take care of it forever, really, just forever. And depending on where you get it on your body and the density of sensory receptors, it can be either really, really painful or less painful associated with that. I have a friend who had dreadlocks, and when she shaved all her dreadlocks off, she got tattoos all over her skull, and she just called me screaming. And I'm like, I told you, dummy. Just radiating through her cranium, and I was like, oh, that was a terrible idea. It's really cute, but now she has hair again, and she's just got like hundreds of dollars that are like under there. Okay, so that's tattoos. And then you want to hear something grody?
Yeah.
Of course you do. Okay, I was going to tell you anyway, so don't say no. Okay, so we know that in other mammals, and you can also use reptiles, the dermis is tanned to form leather. And the tanning process, we're not particularly interested in it, but just so you know, this is kind of how it goes, is that you, of course, have skinned something, and then you've got the dermis. And if you were, for instance, you're going to remove the hair, right? You're going to wash it really carefully to make sure that you got all of the other kinds of tissue off of it. And then you're going to moisturize it, usually with something called tannins, hence the name, right? And stretch it and moisturize it and stretch it. And then you can actually make leather out of it, and the leather has the same characteristics of dense irregular connective tissue, right? So, not surprisingly, because humans don't really like other humans that much, and for other reasons, humans have used human skin to form leather. Yeah, they have. And I was curious, so I went and looked up a bunch of stuff about it. I want to teach you something. Nobody will care until you like. If you have anybody who likes just arcane trivia. So, dermic, human skin, biblio, biblio. What does that mean? Book. Yeah. The art of bookbinding with human skin. You want to see some?
I think we have some
Is
of
this
them.
gonna make it so I never want to read a book again?
Oh.
I already don't like reading, but
I love reading. This just makes me, I wish I could find one. So they're really rare. They do exist. I got to see one when I was in London the last time, which sort of made me curious about this. So let me show you a couple of them.
I think
Okay.
we have a couple of them
At the U?
at the U. And like
Some
the
universities have.
special. Like,
Harvard has one.
Yeah.
I don't know if they, I'm going to go look and see if they have one at the U. I would be really surprised if they have one at the U. There's only a few that are absolutely confirmed. There's like 20 that are confirmed worldwide. Huh? It's before anybody asked. And now they just exist. So, and there's some good stories. You want to hear some good stories about them? So, this one right here. These three have been absolutely confirmed by radioisotope dating and by genetic verification that they are absolutely. The book is bound with human skin. This one, right here. So there's a book collector. His name was Bouland. I don't speak French. Bouland. Bouland. Say it again. There we go. She says it pretty. Okay. So he was a book collector, and two of these three were actually in his collection. This one, I think this one of these is at Harvard. One of them is in Philadelphia, and one of them is in London. This one is a book about female virginity,
Oh my gosh.
which was made and found from the skin of a female patient of a doctor. Here's my question. What the hell are you going to say about female virginity? Couldn't it be a pamphlet? Right? You're like, everybody started out that way. It's not that special. Close. It's a cultural fabrication is what it is, and close the book. But this was in the 1700s. So this was in the 1700s, and this one was made from the skin of a female patient. Not surprisingly. This one right here, you can actually see the pores. Yes. This one, a doctor was caring for a female patient in an asylum, and she died of apoplexy, which is probably a seizure of some sort, right? I would imagine. I mean, they just called everything hysteria and apoplexy back when, and they just said, take cocaine about it, it'll be fine. This is a book exploring the nature of the soul bound in the skin of a female patient who died. And then this one is I think this is a really interesting story. So this one is called the Burke Pocketbook. If I blow it up, you can see it says Burke's Skin Pocketbook. Yes? It was a pocketbook. It was a man's wallet. It was obviously well used. We don't know who had it before we actually found it, but we do know its provenance. We know where it came from because this is an interesting story, interesting historical story. Okay, so there's two dudes, and I think it was 17th century, but it may have been 18th century because I suck at history because I went to school in Texas, so whatever. If you want to know about the Alamo, I can tell you about that all damn day long. Burke and Hare were two what we call resurrectionists, meaning that they procured bodies for dissection for medical school and surgical theaters, okay? And they did that legally until they didn't have enough supply. And then Burke and Hare killed 16 people.
Oh no.
Yes, they did. This is, they were convicted. They were well, actually Hare was not. And they found out that they were actually killing people because one of the surgeons who was procuring the body knew the person and knew that they were recently alive, and they figured out what WTF's going on. And so Burke and Hare were apprehended, and Hare, one of the guys, not Burke, turned like state's evidence. They call it king's evidence at that time, right? Basically, like narked on the other one, right? And Burke was convicted of murdering these people. He was publicly hanged, which was common for murderers then. And then he was publicly dissected, which was also part of the penalties for murder at this point. And somebody scooped up some of Burke's skin during the public dissection and then made a pocketbook out of it. So this is where the Burke pocketbook came from. This one, I think, is at Harvard. This is the one that I saw in the Wellcome Museum in London. And then there's one more. I can't remember where that one is. So that's the Burke pocketbook, but I'll show you a little video that shows you a little bit about anthropodermic bibliophagy.
When and where did you say that was from?
Burke? I think 1800s, I can't quite remember. You can find it really easily. It's just look up the Burke pocketbook and you can see all of the interesting information about it. So this is from the Mutter Museum. I've never been to this one. It's in Philadelphia. I've never been to Philadelphia. I'm going to go sometime. So, and this is one of the museum curators talking about their human skin book. I want to see this too. What the hell's going on there, man? Like,
always nice to get absorbed in a good book. This one is a book from the 18th century, and it's speculations on impregnation in the human female. It's an exploration of how women get pregnant and what happens during pregnancy. Well, that's interesting enough, but this book has another life. In the 1880s, a physician took skin from the thigh of a woman named Mary L. and made it into the cover of this book. This book is bound in human skin. Librarians call it anthropodermic book. And we know from the note that the physician processed the skin in a chamber pot in the hospital in order to make the skin for the binding. But you don't have to lose your skin in order to come down and visit us at the Mutter Museum.
Okay.
Really not that long ago.
I know, right? Yeah. No, I mean they did it like they thought it made the books like a curiosity and a collector's item, and it did. Yeah. Yeah. So anyway, I know you wanted to know that. Maybe you didn't, but I wanted to tell you that, so I did. Okay. So then we are moving on to the subcutaneous layer, and the subcutaneous layer is here. And the subcutaneous layer is made of a combination of two kinds of tissues, which is what? Not dense or regular. Areolar and adipose, yeah. And greater and lesser amounts of adipose. It's not part of the skin proper. So the skin is dermis and epidermis. And this is not part of the skin. It's deep to the skin. And about 50% of your body fat is stored there, but it's really stored differently in high estrogen versus high testosterone situations. So one of the things that happens, of course, at puberty is if you look at two 7-year-olds, right? Their silhouette, male, female, is almost the same, right? But now look at two 17-year-olds, right? And you see that their silhouette changes. And a lot of that is body fat deposition, because what estrogen does is it makes you deposit body fat here and not so much in the middle. So you end up getting sort of that shape. And then high levels of testosterone make you deposit very little body fat in the legs, but most of it, if you're going to deposit body fat, goes from here up. So it gives you that kind of shape. And that evolutionarily, by the way, is the universal signal for sexually viable. Yeah? So interestingly, it has some cultural implications. So heterosexual men, there is almost a universal preference for a certain waist to hip ratio in women. And you can manipulate photos and have them assess, right? Swipe left, swipe right, but only sciencey. And it's about 0.7. So the waist is about 70% of the hips. Even it's pretty consistent across cultures. And some cultures heterosexual men prefer women a little heavier, and some cultures they prefer women a little thinner, right, overall. But the 0.7 is relatively solid. It's almost universal as far as sexual attraction. And men don't know that they like that, right? They just do. Evolution says she's reproductively viable. That's what it says, right? And with women, interestingly, heterosexual women, it's not as if we actually are really motivated by the same thing. So they've done the same sort of assessment in which, like, I changed the look of this male so that he has the body fat deposition that makes him look reproductively viable. Women don't seem to care about that very much, but they do tend to care about the look in a man's face, heterosexual women, that says that he has a decent amount of testosterone. So a relatively strong jawline, relatively strong zygomatic arches, and a brow ridge. If you manipulate that, women tend to assess that man as more attractive unless they are on hormonal birth control. Women on hormonal birth control prefer men with less testosterone and more of the indications of a nurturing personality. So, yes. Isn't it fascinating? Yeah. So you need to see, before you marry him, you need to see whether you find him attractive both ways. Right? You have to go off birth control. Women off birth control tend to prefer more testosterone-y dudes. Right? It's really like who you want. It's kill, bang, marry, right? We're not killing anybody, but who you want to bang and who you want to marry are not necessarily the same person. Yeah, kill, bang, marry, yeah, right? We're not killing anybody, but just evolution, and it gets even more dramatic when you're ovulating. When you are ovulating, you tend to prefer very, very testosterone-y men overall. And I hate to tell you, but when you ovulate, your body really, really prefers variability. Evolution prefers variability. You are way, way, way more likely to get pregnant if you are cheating.
Wait, really?
Yes.
Wait.
Why is that? Think evolution. Think evolution. Think evolution. You've been having the same sperm coming to visit
This is awesome.
5 years.
Let's go off.
You decide this ain't working for me. Ovulation says, hey, he's really testosterone-y, and you say, oh, just this once. And then you get a junior that is not junior. Because your body really likes genetic variability because evolution really likes genetic variability. You are way, way more likely to get pregnant when you are cheating, when it is not someone you have been with before. Isn't that interesting? Yeah, dang. So if you're wondering after the fact whether it's junior or not junior, go ahead and call him junior and hope for the best, but stats say no. So yes, very interesting evolutionary stuff. They've looked at it in humans. They've looked at it in other mammals. Your body likes variability. It really wants to make a new baby. You're like, I already made a couple of those. Let's make a new one.
You always heard that, like the one time, it's like, how did that happen once? That makes sense.
Yeah, it's evolution. Doesn't it make sense to you guys, evolutionarily, right? I mean, I know it's offensive for all of us, but just so you know, sex is just an arms race anyway. So, okay. So, and all of you people who are not heterosexual are going, damn, this is awesome. You're like, hell yeah. Okay, so of course, the subcutaneous layer is also very vascular. It's a common site for injection, so you guys know that. So the last things that we're going to do are just integument repair and regeneration, talk about scar formation, and then a little bit about aging and a little bit about burns, and then we'll move to the nervous system. Okay, so what happens to your skin? This is also true for other tissues to greater and lesser degrees, but what happens to your skin when you damage it? So when you damage your skin, there are two possibilities for ways of healing. Plan A would be regeneration, meaning every cell that I lost, I replaced it with that type of cell. So over time, I wouldn't see any change, right? And you've, of course, done that, right? You've had skin damage that ended up with no appreciable amount, right, of visible change later. And then plan B is I can't replace it with the same types of cells, but I need to plug the hole, and that's called fibrosis, which is scar tissue formation. Does that make sense? Okay, go ahead.
Is that like, do you see that a lot with burn?
Yes,
Okay.
and I'll talk about why.
Okay.
Okay, so regeneration would be ideal if you could do that, and you always want to do that. But note, regeneration is relatively slow, and sometimes it's more important to plug the hole than what you plug it with. Does that make sense?
Yeah.
Okay, because of the likelihood of infection, for instance. So fibrosis is faster than regeneration, right? And in addition, in tissues that are amitotic, regeneration is not a possibility, right? So I can't, for instance, if I damage cardiac muscle cells, they're amitotic, I'm not getting them back. The only possibility is to fill the damn hole with the heart's equivalent of connective tissue, which is scar tissue, right? So now we will talk about fibrosis. So what happens with fibrosis is this process right here in which after damage occurs, first thing that occurs is inflammation, and inflammation is the increase of blood flow to the area. Why would you want more blood flow to the area? Because it brings white blood cells, right? And the white blood cells are both going to scoop up whatever damaged tissue is there, and they are also going to fight off any potential infections that occur. So the cut vessel will bleed into the wound. And then on the top, so what I should have is, if this is skin, on the bottom I should have an increase in blood flow, but on the top I should be forming a clot. A clot that occurs on the skin is a scab. Yeah? So I want more blood flow, but I don't want to lose it outside the body. Does that make sense to you? Okay. And then overlapping with this, what I need is I need to start making some fibers to start knitting that thing together from the bottom to the top. And so the mesenchymal cells that are always hanging around blood vessels will start to differentiate into fibroblasts. And then the fibroblasts are going to make what kind of fibers? What would you make? Start knitting that thing together. Yeah, they're going to start to make collagen fibers from the bottom to the top. So now what I have is I have a really vascular area because lots of blood flow. By the way, you actually sprout more capillaries into the area as well because it's kind of energetically and nutrient expensive to do this. So what I'm going to do is I'm going to have this area where I've got a whole bunch of collagen and a bunch of blood flow to the area, right? But what I did is I spackled it, right? but I lost the original functionality. So punch a hole in your drywall like my brother did when he was a teenager, right? And what happens? It was because I kept coming into his room and just closed the door and punched it through. We tortured one another. There were like salamanders thrown into the bathtub.
Oh my God.
I would answer the phone and I would ask like 5 girls' names just to see if like, is this Susie? Is this you? So we tortured one another. So anyway, he punched a hole. So what happens is you lost the original functionality. So think about your scars. Are your scars usually pigmented the same as the tissue around them? No, right? Do they usually grow hair? No. Do they usually sweat or produce oil in the same way? No. And how about elasticity? Are they usually as elastic as the tissue around them? No. So what happens is you have lost functionality in that region, but it's okay, you plugged the hole. It's not plan A, right? But you ended up plugging the hole. So what's going to happen eventually is this is going to fall off. In the initial stages, it's not done yet, not baked yet, so it's still that sort of pink fragile tissue with a lot of collagen, not complete yet, and a lot of capillaries. That's called granulation tissue. It's pink, it's relatively delicate, it's easy to redamage. But then if you give it a long time, weeks, depending on how big it is, what you will end up with is the capillaries will atrophy and move out. The scar is usually less vascular than the tissue around it, so a lot of times it's paler. It's going to have a little bit of elasticity, but usually not as much as the tissue around it. And the last thing that happens with scars is in the last stage of healing, the fibers sort of pull the edges together, and that tightens the scar. Have you ever felt that occur? When a scar heals, it tightens. And that's all well and good on the skin unless it happens at a joint, which is problematic for burn victims, right? Or this whole process also occurs when things get damaged internally. And if, for instance, your fallopian tube was the thing that was damaged and had fibrosis, the pulling it closed could actually kink the fallopian tube and cause infertility problems. That make sense? Okay. So for instance, like with pelvic inflammatory disease in women, one of the biggest causes of infertility in women is pelvic inflammatory disease. What generally happens is she got some kind of infection in the reproductive tract. It may have been asymptomatic, had no idea. But then it makes its way up into the fallopian tube, right? And as the healing process of fibrosis goes on, it kinks the tube. If you kink a single tube, you reduce fertility by about 50%, because usually it's 50% one and 50% the other, right? One of the many, many reasons that we will talk about this semester to sort of protect the integrity of your reproductive system, right? So that you're not just, for instance, using protection because you don't want to get pregnant, you're also using protection because you would like to choose fertility or not when the time comes. So you have to protect it long before you think. And by the way, when men have STIs, they're usually asymptomatic. Most STIs are asymptomatic in women, so it's not like you could tell that the male actually had. And they're usually symptomatic in women, but not always. So that is the process of fibrosis. Okay, so now why would you do fibrosis? So like I said, you would do fibrosis either because the wound was so big. So think about getting a scar on the surface of your skin. Is the scar usually as big as the wound was? No, it's usually smaller. And why is that? Because regeneration was occurring at the edges, and then you're like, oh crap, this isn't fast enough, and then you spackle in the middle, right? Does that make sense? So the scar is usually going to be smaller than the wound because you did some degree of regeneration. So one of the reasons you would do this is because the wound is too big to wait for regeneration to occur in the whole thing. And the other reason is that some cells are not mitotic anymore, and so regeneration is not a possibility. So here are the 3 categories of cells based on how likely they are to go through mitosis. The category of cells called labile cells are undergoing mitosis all the time. So the likelihood of regeneration with those is really high. So for instance, the mucous membrane inside your mouth, right? You damage it, you're probably going to completely regenerate, right? Your skin, more regeneration than fibrosis. You really would only do fibrosis if you had a really relatively big wound. Or like your bone marrow, your red bone marrow, you can donate that because you'll just make some more. It's always going through cell division. That makes sense? Okay. Then the next category of cells are called stable cells. Stable cells don't regenerate all the time, but they remember how. This is like most of the glands, most of your endocrine, exocrine glands, your liver, your pancreas. All of those aren't dividing all the time, but they can, which is why, I don't know if you guys know this, but you can do living liver donations now instead of just cadaver donations for liver or body or organ donations. So if I had someone who I adored and they were a tissue match, I could actually give them a lobe of my liver, and within about 2 years, I would have regenerated the lobe. And within about 3 years, assuming that they didn't have rejection, they probably would have filled almost the entire space that the liver takes up, right? Yeah, so you can do living liver donations now. Okay, and then the ones that you want to protect at all costs are called the permanent cells, because permanent cells are amitotic, and the primary permanent cells you're going to be concerned about are cardiac muscle. After you have a heart attack and those cells die, they're dead. And you are more likely to have a heart attack in the future because you've got fewer cells doing the same job, right? Okay, you are not getting them back, but you don't leave a hole in the heart, so what do? Is you spackle, right, where those go. And then the other one is, of course, neurons. If you have a stroke and the tissue dies, the tissue is gone. You don't get it back. Luckily, we have what's called neuroplasticity, meaning that a lot of your neurons can pick up the slack for the ones that died, especially when you're young. They're really good at doing that. So neurons and cardiac muscle cells. Okay, so, and then last two things is just what about skin throughout your life? What can you expect of your skin after your 20s? Your skin starts to age appreciably in your 30s, definitely. But you can speed it up, do it by doing some things to it. We'll talk about that in just a minute. So as you age, one of the things that occurs is that dermatitis becomes more common. You get more irritations and inflammation of the skin than you did when you were younger. And then your oil and your sweat gland secretion slows down. The oil gland secretion slowing down means that your skin will get drier as you age. Now, the only good thing about that part of the story is if you actually struggled in adolescence with oily skin, you are probably going to age better than most people. You're going to get some props from that later, okay? Which is good, right? Because you want to be pretty when you're 40, because everybody was pretty when they were 20. Everybody, right? They may not have thought it, but if you look back and you go, damn, I should have appreciated that. Okay, so you got fewer Langerhans cells. Who remembers what those are? Yeah, those are immune cells. Since you have fewer Langerhans cells, you're more likely to contract an infection when you actually get exposed to something. Okay, your collagen and your elastic will decrease. Really what happens is you break them down and you just don't replenish them as readily. So what happens, of course, is you start getting wrinkles, right? Okay, and your skin starts to sag because of the loss of collagen, okay? And that happens to all of us. And then your melanocytes decrease, both in number and also in activity. And what does that mean? If your melanocytes decrease, what's going to happen to the color of your skin as you age? It tends to get lighter. I don't know if you saw this. She died yesterday. It was one of the mathematicians who worked on the moon landing. Yeah, she worked at NASA on the moon landing. She was 101. She was African American, so it took decades before she actually got credit for her work. But she was in the movie Hidden Figures, right? Or her character was in the movie Hidden Figures. But the thing that I noticed yesterday, because I was about to teach this, is that they show pictures of her when she's young African American, and they showed pictures of her at 101, and her skin was really much fairer at 101, right? So your melanocyte activity decreases as you age. And this is assuming you're not tanning, right? I'm not saying that you won't tan. I'm saying that the inherent skin color decreases, gets lighter as you age. You also reduce sweat production, which makes thermoregulation a little harder. Your vitamin D production decreases, which can impact your bone mass. Your body fat deposition will change again. So when your body fat deposition initially changed, for instance, in women, what happened is you went from being this shape when you were 7 to this shape when you were 17. You're going to go right back to that shape. And that is evolution being a jackass and trying to signal to males that you are no longer reproductively viable. Yeah? That's what it's trying to signal to males, because that happens at menopause. And there are women who can battle against that, but you are not going to have the figure you had at 30 at 60 without working your butt off. It's just really difficult because evolution is trying to tell everybody, don't spread your seed here, no point, right? Okay. And then since your skin thins, two things happen. You're more susceptible to temperature change. You're also more susceptible to skin damage. And again, we talked about that. If you've worked in an old folks home, right, you can move them around. If you're not careful, you can actually rip their skin just with your hands. And then the last thing to talk about skin and aging is the sun. Just talk about the sun and aging. And so the 90% rule is that dermatologists attribute 90% of skin aging due to sun exposure. 90%. And I've got some things that will prove that to you in just a second. So most aging is due to sun exposure. So let's look at some examples. Well, first, let's talk about just aging in dark skin versus light skin people. Not tanning, just talk about your inherent skin color, right? So have you noticed any difference, just anecdotally, in how a person with light skin ages compared to a person with dark skin? It's really, really dramatic, right? You can have somebody with dark skin who can look 20 years younger than somebody with light skin. And that is simply because what UV radiation does is breaks down your collagen and elastic fibers. But if you have the inherent protection from UV radiation, then your collagen and elastic fibers are more protected, right? And so you get less sagging and less wrinkling associated with dark skin. Does that make sense? Okay. So now we can look at some examples, and here's one right here. So if you really want to see what happens with aging, these two women are identical twins. Every year there's a twin convention in Twinsburg, one of those I states, Ohio, Iowa, Idaho, I don't know, an I state for sure. Where is it? Illinois? I don't know. It's got an I in it. Anyway, there's a twin convention, and some brilliant dermatologists decided that they would go up there and they would actually interview people. And so we have, of course, identical genetics, right? And in this particular pair, I know they were raised together, so they had the same environment, at least during their developmental stages, right? But the difference between these two, there's actually two differences. One is lots and lots of sun exposure in adulthood. And you can see it's super duper dramatic what sun exposure does. You can see both the sunspots, you can see the wrinkling, you can see the loss of elasticity. And the other thing that helps the woman on the left, twin A, is that she's about 15 pounds heavier. And once you get into your 40s, a little bit of subcutaneous fat reduces the appearance of aging. When you're in your 20s, it actually makes you look older. When you're in your 40s, it actually makes you look younger. So a little bit of subcutaneous fat. So these two are twins. And then this guy is an even better study because he is the same damn person who is a truck driver.
Oh.
Is that not dramatic enough for you to wear some sunscreen? Right? Because even those of us who are not very vain are like, oh, that's not okay. Right? I mean, that's not me. I'm vain as hell. But other people, right? So do you see this dramatic change, the loss of elasticity and the wrinkling associated with the sun exposure? Yeah? So and look at all the sagging. This eye is measurably lower than the other eye. I mean, I don't know what he looked like originally, but I would imagine a lot of that is due to sun exposure. And then just to make you really take this seriously, how about that? You like how that ages? Yeah. Look at the little boy's face. I don't know what he's doing. He probably just ate a lemon, but so
Straight up in the sun.
she is in the sun. I don't think she has sunscreen on. Do you?
No.
Mm-mm.
Obviously not.
Nope.
God, that'd be so bad.
That's crazy, right? That's what living in Florida will do to you. So do we all feel a little prettier than we did just a minute ago? Yeah, right? Okay. You just go out today and you realize this is as hot as you're ever going to get and act like you own the damn world. That's what you need to do. Okay? Okay. And then last little tidbit is just to talk about burns. Okay. So what do burns, first, second, third degree burns have to do with like all of the stuff that we've been learning? So a first degree burn just damages the epidermis and irritates the dermis. Okay. So typically speaking, what are the symptoms associated with a first-degree burn? Not bubbling, that's second. It's just redness, right? So if we look at first-degree burns, where are we? I lost it. So this would be a first-degree burn, yeah? Okay, now, a first-degree sunburn has risks associated with it that, for instance, a steam burn or a chemical burn or a heat burn does not. And what are the risks associated with a sunburn? Yeah, the UV radiation and cancer risks. So while they seem minor, you know that you have actually damaged the, or had UV actually hit the upper level of the dermis, right? Does that make sense? And went all the way to the bottom, right, of the epidermis by definition, or else you wouldn't have any excess blood flow going there. So that's a first-degree burn. A second-degree burn is when you damage the entire epidermis and the upper portion of the dermis. And generally speaking, you are going to have blistering associated with it. All a blister is when you have inflammatory fluid that pulls the epidermis away from the dermis. And since the epidermis is pulled away from the dermis, does the top of the blister have any sensory capacity? No, because you pulled it away from the nerve endings, right? If you take off the top, the dermis still feels, right? Okay, so this means that you damage the entire epidermis, and you know you did damage to the dermis, right? At least the upper portion of the dermis. And then the third-degree burn is this guy right here. Third-degree burn damages the entire epidermis and the entire dermis. Fibrosis is really, really likely with third-degree burns because you have to plug the holes faster than regeneration can occur, right? And so with a third-degree burn, you can, for instance, damage all the way down and crackle the fat underneath with third-degree burn. So since regeneration is, what? I have no idea what that's from. They didn't tell me. Mm-mm. It looks like something spilled. Yeah? Because her clothing, or his clothing, was on here. It's probably not sun exposure. Okay, so what do we do? By the way, when they try to figure out how much was burned, they don't measure because ow. They just know that, for instance, in an adult, a limb is like, I don't know, I can't remember, it's like 8%, 16%, torso is X%, head is X%. You have to recalculate it based on children because, of course, their head is ginormous compared to their body, right? But they don't measure; they just estimate based on the body proportions of a normal human. So, what do you do to treat burns? In addition to all the other things, of course, you have to thermoregulate them. You have to keep temperature and fluid constant. But how do you help burns to heal with as little fibrosis as possible? Well, there's a possibility of tissue transplantation. The ideal tissue transplantation would be from me to me. That's called an autograft. So if, for instance, I have a burn here and I can take chunks of tissue from here and here, each little space might be small enough that you don't really have a lot of fibrosis, right? And you use it to fill the bigger space. Does that make sense? Okay. So that's called an autograft when it's from me to me. When it's from another human to me, it's called an allograft, another human. And the primary sources of allografts are two primary sources of allografts. First off, we use cadaver skin, but it's actually less gross than you think. It's actually really kind of interesting. What we want to do is we want to reduce the amount that has the space so that we make fibrosis less likely. So with cadaver skin, what they do is if you are, for instance, an organ donor, this is one of the things that you might be donating. And they actually take it and they take a little square of skin and they run it through a planer so that it's really, really thin so you get tons more skin. And then they use that as a space filler to reduce fibrosis so that you do regeneration at the edge and then you take that chunk out and you put a smaller chunk in. More regeneration at the edge, take that chunk out and put a smaller. Isn't that kind of cool? It's clever, right? They do the same thing with foreskins of little boys who were circumcised. You don't tell them not to, so what they do is they just donate that tissue. And again, the foreskin doesn't go in, but it's really, really regenerative tissue. So what they do is they grow those foreskin cells up in culture and make little bitty layers so that you can stick it in, pull it out, stick it in, pull it out to reduce fibrosis again. It's actually pretty clever. And then the other thing is artificial skin. I'll come back to heterografts. We don't do those very often in this country. Artificial skin is, I can do the same kind of thing, but add a little more connective tissue underneath. If I use on the bottom side, I use an artificial mesh. Like the mesh is like pretending it's dermis, right? An artificial mesh that will disintegrate and be absorbed after it has filled the space. And then they actually spray skin cells on top of the artificial mesh. They have them aerosolized and they spray them on top of the artificial mesh. Yeah. And then that's like the placeholder for your skin to grow around it. And that's how artificial skin kind of works. And then the other one we don't really use in this country is heterografts. Sometimes they have to use the skin of another organism, usually a pig. Those are way, way more likely to be rejected by your immune system. Okay, but they have been used, historically speaking. It's just that we have a lot of other techniques here in this country that we do. Go ahead.
So what, I mean, obviously, like, if you're donating blood, there has to be a match with skin. Is there a specific, what determines?
Well, the good part about skin is that it's not vascular. And the whole antigen-antibody thing is far less of an issue with skin than it is with other tissues. The skin rejection can and does occur, especially if you actually give the whole dermis. But if you're primarily just giving the epidermal cells, then the likelihood of rejection is relatively low.
Okay.
Does that make sense? It's not zero, but it's relatively low. So we can actually get around them, especially since a lot of the cells don't end up being part of you permanently anyway. So it's kind of interesting. That's one of the ways that they treat it. Okay, everybody good? Skin? Okay. All right, so now we are going to start. What is that? I really need to know. That little blob of, what is it? Is it like one of those, what's it called when you have like a fetus that doesn't develop and it just develops? There's a name for that. I don't know what the hell it's called. I want to know what that is. Yeah, but there, yeah. Huh? What'd you say? Of course there is.
Like chimera?
Well, chimera is when your genetics is shared, yeah. But some people actually insist a partially developed, I don't know what that is. Parasitic something? Okay. I don't know. I'm going to look it up. I'm going to go look at the Mutter Museum and find some more dumb puns and figure out what the hell that thing is. Okay, so now we start my favorite topic. Hopefully you guys will like it as well. It is not the easiest topic. It's the hardest one in anatomy, but not this first part. It's the brain that's actually the hardest. Nervous system. And nervous system, I think it's challenging for students. I'm good at teaching it now. I didn't used to be, but I've gotten way better at teaching it now. One of the reasons it's hard for students is, first of all, we don't know what to leave out with the nervous system because it all seems so important, right? So there are literally 6 nervous system chapters. So how do you figure out what to study? Well, you don't have to, because I did, right? I narrowed it for you based on A, cool stuff, because we liked cool stuff. And then B, what I know you're going to need for physiology, and C, what the clinical programs tell me that you need. And then D, I just did this one, and I'm going to change your brain notes, not these. The first set of notes is fine. I'm part of a neuroscience research group, and I asked them, based on their research, what are the brain structures that every single person going into clinical applications needs to know? And now I'm going to revise my set of notes on that. But that's the next set of notes. So it's difficult to figure out like how to narrow it down. And then the other thing is it's less familiar to people. The terminology isn't very familiar because, for instance, you were a human. You learned that this was a humerus at some point, right? You're like, this is a deltoid. That's fine. That's skin, right? I got this. But most of the terminology of the nervous system is not terminology that people use. Even, for instance, if you take high school anatomy, they don't teach very much nervous system stuff because it's really, really hard to simplify. So expect for a learning curve to exist for terminology. That's what I'm saying, okay? But it's okay, because we've got some time before you actually have your test over this material. Your test is the week after your lab practical, okay? So you still got some time, and it just covers muscle, which is also on your lab practical, skin, which is also on your lab practical, and then this stuff, which you never even learned in lab anyway. So that's what's going to cover. And this isn't the hardest part. But please do stop me when you get confused instead of 15 minutes after, because somebody else probably has that question too. Okay, so let's start with just the basic sort of intro functionality to the nervous system before we get into any detail. So big picture understanding what the nervous system does. Of course, this is an oversimplification, but we'll work with it. So the nervous system primarily is doing a combination of these three things. What it is going to do is it's going to be one of the body's two control centers. It's really communicating, right? But it's your fast communication from one point to the other. The endocrine system is your slow communication from one point to the other. Why is the endocrine system slow? How do hormones get from point A to point B? Bloodstream, exactly, right? Collectively, those two are really trying together the vast majority of the time to maintain homeostasis, right? So let's talk about a really simple example of how the nervous system does this. And we've done it before, but we're going to add a lot more detail to it. So what the nervous system is really trying to do is a combination of these three things to generally maintain homeostasis. So again, if I put my hand on a hot stove, three things need to occur to maintain homeostasis. I need to carry the information in. That's called sensation. Everybody with me? The second one is where it can get really complicated, but let's keep it simple for just a second. Integration is what happens inside the brain or the spinal cord to assess and figure out the response necessary. So, for instance, hand on hot stove, information in, that's sensation. Information processed, that's integration. Meaning what the brain would be doing is saying, hot, identification. How hot, assessment. Too damn hot, move your hand, right? Okay. All of those decisions go on in the central nervous system, and that's integration. Yes? And then the last one is response, or the motor response. So what I have to do now is I have to send out a stimulus to cause the desired response to maintain homeostasis. Yes? So sensation is information in, integration is processing, right? And response is information out, okay? And the nervous system does varying degrees of complexity about all of those kinds of things. Of course, this is a really simple sensation, integration, response, but you deciding who to vote for is a very complex sensation, integration, response, and please do vote.
Yes.
Please do. I don't even care who you vote for, but the old people ain't doing us right. You need to get your asses out and vote, okay? Right, and there's more millennials than there are old people, so if you just vote and vote your little butt off, right, unless you think things are going really well. Do you? Okay. Right. I'm not a boomer or a millennial. I'm a Gen X, so I just make fun of both of you. Okay. So I don't. I actually really like millennials. I do. Okay. So now what we are going to do is we're going to talk about the general organization of the nervous system. So first, you have heard many, many times of nervous tissue referred to as gray matter and white matter. Okay, I want to put that together so that it can attach some significance to both the functionality and to the tissues that you've learned before. So stick with me for a second, ask questions if you get confused. So if we're just talking about the brain, let's start out talking about the brain, but we won't limit it to the brain. When you're looking at the brain, they refer to gray matter and white matter. So let me show you what they're talking about. It ain't gray unless you're dead. Okay, this is a real brain, fresh human brain. And what you are seeing here is the outer portion is more pigmented because first, it has more blood flow. So it's actually pink matter when you're alive. When you're dead, it's no longer pink, so it's gray matter. Okay, that's where most of your neuron cell bodies are. Okay, so I want you to think about that as doing most of the processing. Yes? So the neurons are, to oversimplify, kind of your little processors or decision makers. Okay, so the gray matter is here. That's not the only place that gray matter exists, but I want you to have a visual of it. Okay, and then the white matter, stick with me here. The white matter is the wiring between the processors. Okay, so let me give you an example of why you need wiring between the processors. Do you want to see what the wiring looks like with a space-filling model that somebody made that was super cool? Okay, so if you pull off all of this, somebody did an fMRI to look at how all of the wiring is set up in the brain and made a model out of it, and it's the most beautiful thing. I would pay so much money for it. This is the wiring within the brain. That is the white matter within the brain. Okay? All right. So, gray matter, thinking, processing, decision making. But the brain is specialized, the different portions. We're not going to talk about the specialization of the brain yet. I'm just trying to get you a grasp on what we're talking about. So I want you to imagine that I want to talk to Anthony about this lab manual, right? what's going to happen is I'm going to need to use multiple portions of my brain to do it. Okay, the visual identification is going to go on in the back of my brain, in the occipital lobe. But the speech processing goes on in the left portion, the left temporal lobe. So what I'm going to have to do is I'm going to have to use those together, right, in order to, for instance, say Anthony, and I have to recognize that it's an Anthony too, right? Okay, Anthony, this is a lab manual. What is going on is identification, going on back here, and then processing of the actual pattern of sounds and speech. There's actually even another portion up here that actually produces the motor response necessary. And so what I have to do is I have to use all of these little specialized processors, but they have to communicate to one another via the wiring, the white matter of the brain. Does that make sense? Okay, good? Okay, so the gray matter is really kind of the decision makers, the neurons themselves, and the white matter is primarily myelinated axons going from one to the other. And some of them go from back to front, yeah? Some of them go from side to side. Some of them go within a region, front to back right here, side to side, up to down, right? So there's a whole bunch of different white matter within the brain. Does that make sense? Okay, so here, what would happen, this is the back portion of the brain, the occipital lobe, because that's the cerebellum right here. What would have happened, let's see, left, right. What would have happened is I identified it, and I identified Anthony, took it over to the left temporal lobe to figure out how to say lab manual and Anthony, right? And then there's actually a portion up here where I actually use to actually force the motor speech that causes that. And I have to connect all of that with wiring. Go ahead.
I don't know if I'm simplifying this too much, but is the white matter kind of like communication system within the brain, and then the gray matter is what sends communications throughout the body?
The gray matter are the cells that actually decide. It's not conscious, but they are actually deciding to send the signals. And then the signals move down the white matter. So I kind of think about the white matter like a coaxial cable, right? Like a wire made of wires, right? It's a bundle. Does that make sense? Yes? Okay. So that's how the white matter actually works within the brain. And by the way, we have white matter in the brain and the spinal cord, and then we're going to have more white matter outside of the brain and the spinal cord. So somebody had a question, and I'll answer that. Go ahead.
So with the white matter, is it mainly just communication between what?
Between anything. So what I'm going to do, so let's put this back together with what the example that we talked about. And by the way, this is hard to grasp. So just ask questions until you freaking get it. You're 15 minutes ahead of the other class anyway. So so I want you to imagine again that what is going to happen is I'm going to put my hand on a hot stove and respond to it. So big picture. So we just looked at the brain, and the brain was gray matter and white matter. Yes? Okay. But then what I have is I have nervous tissue running out from the brain and the spinal cord. So the brain and the spinal cord are considered the central nervous system. And for reasons that I hope will become obvious in just a second, that's where almost all the neuron cell bodies are. They're in the brain and the spinal cord. So what do I need to go out to, for instance, sensory receptors and muscles, right? So I can put my hand on a hot stove. The information is going to come in, get processed in the gray matter, move through the white matter of the brain to get it to the right place, right? And then I'm going to send it back out. What do you think most of the peripheral nervous system is? White matter. Yes. And why is that? Because A, it's mostly wiring, and B, do you want your neurons outside? Why not? They're not very protected out there. So the vast majority of your neuron cell bodies are sequestered within the brain and the spinal cord, where not only are they encased in bone, they also have 3 layers of membranes around them called meninges, and they have protected fluid called cerebrospinal fluid around them. Yes? So what is out here? Primarily just coaxial cables. Let me show you what it looks like. So if I take a cross section of one of these little buggers, right, what does it actually look like? It looks like this. Notice at the very top you see a single axon that's myelinated. What's special about a nerve, stick with me, versus the white matter in the brain is that a nerve needs layer after layer of connective tissue protecting it. Do you need that when you're actually in the brain?
No.
No, right? You just need bundles of myelinated axons. Out here, we need myelinated axons wrapped in epineurium, perineurium, and endoneurium. Yeah? And that's what makes it a nerve. Are we okay with that? Okay? Kind of understand the big picture. Now do you understand why your high school teacher doesn't teach nervous system? Because it's freaking hard to teach nervous system, right? It's hard. Okay, so gray matter, white matter. So the white matter is mostly myelinated axons. It's wiring, yes? And the gray matter is the cell bodies of neurons and a whole bunch of unmyelinated axons. It's usually pink in living brains, yes? So it's not gray until it's dead. The ones you see in lab are going to be preserved, so it's going to be gray in those, right? Okay. And so now let's just look at the basic structural organization of the nervous system before we go any further. So central versus peripheral nervous system, just making sure you understand the terminology. The central nervous system is down the center. It's only the brain and the spinal cord. It's very, very special because that's where above 97% of your neuron cell bodies are. And it's mostly doing what? You guys tell me. Sensation, response, or integration? It's mostly doing integration, right? Because sensation is going to come up through these, yeah? Integration is going to happen in there, and then the response is going to go back out through there. Does that make sense? Okay, so that's the central nervous system. The peripheral nervous system is every other nervous tissue in the body except brain and spinal cord. And it is primarily composed, almost exclusively, there's a couple of little exceptions we'll learn in just a little bit, primarily composed of nerves. And what is a nerve? A nerve is a coaxial cable made of myelinated axons and all of the connective tissue that protects them. Yeah? Okay? And it's really just wiring. And so, again, if you had to guess, the peripheral nervous system mostly does, choose any that's appropriate or all, sensation, integration, or response. Sensation and response, hardly any integration. Yes? Okay? Does that make sense? So the information comes in. Go ahead.
Where would it do any integration?
There's a couple little, this is hard to understand, but I'll just tell you for later. There's a couple of little blobs of actually neurons in the peripheral nervous system. They're right next to the spinal cord. So it does a tiny little bit of integration. Go ahead.
Sorry, so you said the peripheral nervous system does sensation and integration?
No, sensation and response. Hardly any integration. Does that make sense?
Yes.
Is it logical? Okay. All right. Okay. So, and you know the answer to this, right? Central nervous system has the brain located in the what cavity? Cranial. Spinal cord located in the spinal or vertebral. And if you look at a chunk of brain, I want to talk about what's in there. And then, oops, balls. Okay. So, if you look at a chunk of brain, what you are going to find is this is neurons, right? And glial cells that are not myelinating glial cells, okay? And then this is primarily wiring, okay? So is that okay? Okay. So if I looked at a chunk of that gray matter, I don't know why they pulled it from there. It's better from here. What you would see is a whole bunch of neurons and their support staff. Yes? Okay. Neurons and their support staff. I'm not saying there's no myelin in the gray matter, but the white matter is almost all myelin axons. Does that make sense? So back to our metaphor that we used before to understand why neurons need a support staff. These are two neurons, right? This is the Olsen twins, right? They don't know how to do a damn thing without their support staff, right? So this guy makes sure that they get the nutrients they need and brings them coffee, right? This little guy right here is their ninja security officer. He whoops up if any kind of pathogen gets in there. This one holds their cell phone so they can get their information in and out, right? And then there's another one that I can't come up with a metaphor for. Okay, does that make sense? So that's what you would see if you, for instance, took a chunk of. Now, when you looked at a histology slide from nervous tissue before, they just called everything glial cells. But there's actually 4 different kinds of glial cells in the central nervous system, and there's 2 types in the peripheral nervous system. Okay? Is that okay? So it's more complicated than you guys learned before. Questions? No questions? No? You sure? Go ahead.
Where that slide, where was that located?
It would be coming from anywhere in this gray matter. I don't know why they took it from there because it's not white matter, but anywhere in the gray matter you would see neuron cell bodies and all of these supporting cells. It's just kind of like if they chose like an unfamiliar place. There's gray matter, by the way, the gray matter, last little thing and then I'll let you guys go for today. here is something that I find both fascinating and super duper humbling. The nervous system is my total favorite, in part because I want you to know that every single sensation that you have has to come to the central nervous system for integration. Has to. But it doesn't have to tell you about it. You are only aware of something if it comes to what we call the cerebral cortex, which is the most superficial, about 4 millimeters of tissue on the outside of your brain. Okay, so if it doesn't go there, you never know it occurred, but your body is still integrating it and responding to it. So for instance, right now you are monitoring your own blood pressure. But since that sensory information goes to your medulla oblongata, you have no idea that you're monitoring it, right? So you're only aware of sensations that go to your cerebral cortex, period. For that matter, you are only in control of responses that come from your cerebral cortex. So you are, for instance, from your medulla oblongata right now causing vasoconstriction or vasodilation or increasing heart rate to regulate your blood pressure, but you have no control over that, right? The only thing that you have control of is what comes from your cerebral cortex, and your cerebral cortex is wired up to a single tissue. And what is that? Skeletal muscle. That is all you can actually control. So there's a whole hell of a lot going on behind the scenes, primarily for maintaining homeostasis, and we have voluntary control over very little of it. Last tidbit, I'll let you guys go. So can you conceptualize now why, when someone, for instance, is still breathing and their heart is still functioning, but we have no indication that their cerebral cortex has any blood flow or electrical activity, for all practical purposes, from our level of knowledge, is that consciousness still alive?
No,
No.
it's just the body function.
It's the body functioning, your cerebral cortex. Everything that is you is in this superficial portion of the brain as far as we've ever discovered. The outermost portion. It is. It's some profound shit, isn't it? Right? Like, if you ever get a chance, well, you won't, probably, unless you work in neuro, to hold a fresh human brain. It's like every single thing that this person has ever thought, been imagined is all like in the palm of your hand. But you do get to hold, not fresh human brains, but we've got a whole bunch of preserved